genetics and fibroids. The most common genetic mutation related to fibroids is the MED12 mutation that occurs in up to 90%. And so what this tells us, again, 45 to 90% is not a good range. So it's telling us the studies were not big enough, we needed meta-analysis, or that there's too much variance within categories. And so for instance, this genetic mutation is most common in women of African descent. And so they are going to have a higher expression of this than other women. And so if you don't control in your study for ethnicity, then you're going to be having these wide ranges. And so basically, there needs to be a better study done or meta-analysis because this range is not helpful in understanding what's going on. What happens with the MED12 mutation is that you have increased collagen expression. Now, African Americans are also at greater risk of throwing keloids. However, the MED12 mutation is not related to keloid formation as we understand it now. Another genetic risk factor for fibroid is overexpression of HMGA2 that is found in about 7.2% of fibroids. And this is what causes the myocytes to change and angiogenesis to occur and throwing more of that extracellular matrix, the collagenous matrix. Also, fumarate hydratase mutations or deficiencies promote growth, but you'll recognize these because you don't expect to see fibroids in younger women. You don't expect to see fibroids in a 20-year-old or a teenager or anything like that. And so if you see fibroids in someone who is young, then you're going to guess that probably they have this mutation. Another gene that can be disrupted is COL4A5 and 4A6. That's in 2% of the fibroids, and it's related to insulin receptors, cell proliferation and angiogenesis, and Alport syndrome. So again, we want to look at the inside of the fibroid and the estrogen receptor A promoter, which is the regulatory region of the DNA that controls expression of the gene that codes for the protein of the estrogen receptor alpha. What we know is that when there is hypomethylation of that promoter, we're going to have overexpression of the receptor, the estrogen receptor estrogen. So by now, most people are most familiar with MTHFR as a reason for hypomethylation. And so many people have already had their gene run for MTHFR. And so the one that is related to methylation is the C677. And so if you have a heterozygous or a homozygous SNP or single nucleotide polymorphism of that gene, you are not going to be as good at methylating. So when they looked at increased risk of fibroids for women who had the MTHFR C677 SNP without controlling for anything, they found that these women had a 43% increased risk. That means that for women who either had the homozygous or heterozygous SNP, and all of those were included: women independent of age, height, weight, body mass index, parity, gravity. All of them were included just straight up. There was a 43% increased risk of having hypomethylation related fibroids if you have the MTHFR C677 SNP. When they controlled for confounders, the ones that I mentioned, age, height, weight, BMI, parity, gravity, and also the heterozygous mutant, that went down to 41%. And so what that tells us is that those are almost the same. And so it tells us that this SNP, whether it is heterozygous or homozygous, is pretty strong at promoting fibroids. And what we know is that if you are heterozygous, that means you get one thing from your mom and one thing from your dad, right? And so these genes, if they're both wild type, that's normal. If you have one wild type and one mutation, that's heterozygous. And that's a 40% reduction in methylation. If you get two SNPs, one from your mom, one from your dad, then that's a 70% reduction in methylation. So that's very, very significant. And so, but either way, 40% even is adequate to hypomethylate significantly. So this is the green one is the methylation cycle. The blue one, they're calling it here the DNA cycle, but generally it's called the folate cycle. And then the pink one is the transsulfuration pathway. So this folate cycle, the kind of bluish purple one, is important for DNA synthesis and DNA methylation. And you can see the MTHFR gene is right here at the very bottom. And so what it's supposed to do is come up right here to MTR and contribute a methyl group so that you can get methionine. And so that is methylating, right? And so if you have a genetic SNP in this MTHFR, you have 40 to 70% less swing up here to help to methylate this methylation cycle. And so that's considerable, right? That then will slow down this methylation cycle because it doesn't have the right building blocks to do its job of DNA repair, gene expression, detox, neurotransmitter synthesis, and hormone regulation. And so MTHFR, SNPs, single nucleotide polymorphisms or mutations, significantly impact methylation, which significantly impacts fibroid